Whenever we are talking about the sample collection, it is very important and has its significance. What was the time for the sample collection in the autopsy that we are carrying out, and we are suspecting poisoning at this level? So we have to keep in our mind the discussion is basically dealt in keeping in our mind that we are suspecting poisoning in some sort of, there's a foul play of poisoning in the death, and now we are dealing with that type of death. So we have that in our mind and the sample collection is being done. What type of fluids we are going to collect from the dead body, what type of tissue samples we are going to send to the chemical pathologist, that is the only thing that we're going to discuss over here in this sample collection regarding the suspected cases of the poisoning over here. So the time of sampling, of course, it is very important, and whenever we are going to send the samples, the labeling that is going to be over there on the test tubes or the bottles or the plastic containers, whatever it is, you have to mention the time of the collection of the samples, the basically the time duration between the time of death and the collection of samples, how much time has been elapsed. If they were the live samples that we were talking about, what was basically the time that these live samples were taken from the patient. And so the information supplied to the laboratory. So whosoever is performing or whosoever is working as the chemical pathologist or the histopathologist, he should have a complete knowledge. Even a telephonic call can be administered at this level to the chemical pathologist, and some details about the person that the autopsy has been carried out, the age, the name, the other particulars regarding the person can be dealt in personally to the chemical pathologist. What is the suspicion of the person who is performing the autopsy, what he wants to look for specifically. If there is some sort of special request that can be made to the chemical pathologist at this level when the samples are being taken and sent to him to the laboratory. And the information supplied to the laboratory is again going to be mentioned in detail over the samples taken. The containers and the preservatives are again going to be looked for over here, and the containers can be specific containers, and the preservatives, for example, for the blood we use fluoride, and that has to be mentioned what kind of preservatives have been added to the containers. Again, anticoagulants can be added to the blood samples just to avoid the coagulation of the blood. So the whole procedure, how is it taken, why are the samples important, they are discussed over here in this section. Now, the correct and the appropriate samples are taken for the toxicological analysis. So whatever suspicions we have in our mind, according to that, we have to assemble these samples collection and the toxicological analysis. Even in the decomposed bodies, if a body is putrefied or we have collected, we can get traces of the poisons. For example, the arsenic poison can be detected even in the graves because it can be accumulated in the soils. So all these samples, if exhumation is being carried out, these samples can be sent to the chemical pathologist, and your suspicions are always over there, and you have to tell him or her that these are the suspicions that I have, and you have to look for this specific kind of poison in these samples. So entomotoxicology, it is another branch of the toxicology in which what happens is it's basically the presence of the maggots, and the maggots can basically if the maggots are not over there, then there are some of the other insects that can help in the detection of the poisoning. And this branch is a newer branch, and it is basically known as the entomotoxicology. It's basically the presence of the insects and how are these insects helpful in determining what kind of poisoning has been there. So the time of sampling is again very important. The shorter the delay between death and the removal of the samples, the better it is, and the easier it is to see what kind of poison was there and what kind of foul play was there, whether it was accidental, whether it was homicidal, or whether it was iatrogenic in nature. So then we have the mortuary refrigeration. So if there is some delay in the performance of the autopsy, the documentations are not there, you just don't have to panic. What you have to do is just put the dead body in the refrigeration, and it is going to slow down the processes and the half-lives of the drugs or the poisons if they are there in the bloodstream. So it is another method of the refrigeration method, is another method of preservation. And the samples are preserved for longer duration by adding of the additives. So it should also be kept in our mind. Now the information supplied to the laboratory. So as you can see over here, even in this diagram, you have to name the person, you have to put on the date, when was the sample collected, what was the time duration, what was the time duration between the death and the collection of the specimens. What are your specific special requests? If any, you can mention them on the labeling of the samples. So the personal details of the deceased are made over here, and then we have the brief details of the symptoms, if any, and the length of the illness can be recorded over here. Then we can add the post-mortem interval, the actual date and the time of sampling, the name, address and the telephone numbers of the pathologist have to be mentioned on this labeling. A list of all the samples provided with an indication of the sampling site for each. So it is very important that we mention over here what were the indications for the sample, what had the pathologist had in his mind when he was taking these samples out. For example, if you are taking the brain samples, you are suspecting some sort of narcotic, some sort of opium, some sort of tranquilizers, poisoning. It can be of an iatrogenic origin. So that all has to be mentioned over here on the labeling of the samples. And then the nature of any preservative in each of the samples. For example, if fluoride is used for blood, it can be mentioned on the test tubes. So a note of the storage conditions, if the preservative is used or if it is not used, it can be mentioned on the bottles. The special risks, if any, for example, if the patient had HIV, hepatitis B or hepatitis C, these contagious diseases have to be mentioned on the labels just for the knowledge of the chemical pathologist and just for his safer side. So the precautions for the continuity of the evidence are again very important. What kind of precautions we are going to take over here? For example, the evidence and the other criteria on the clothing and the external surfaces, they have to be preserved over here whenever carrying out the autopsy. The labeling of the containers, as already mentioned, it has to be a detailed labeling including the particulars of the patient and the special requests, if any, in different cases. Then the pathologist countersign. So the pathologist has to countersign any findings that he had while performing the autopsy. And the labels and the seals have to be exhibited very clearly on the specimens. The correct identification of the specimens is again very important. So we have to name the specimens of which cadaver they belong to. We have to name that specimen. Now, the containers for the toxicological samples are again very important. The container types, it can be a plastic container, it can be test tubes, or it can be glass bottles. So it depends upon different laboratories. They provide you with different sampling kits. Some of the sampling kits, they include different bottles for the tissues and different bottles for the blood and the urine and the bile and the CSF samples. It can be new, cleaned or sterilized, and screw capped containers. So it is usually very important to protect your samples. And the preservatives used for these sampling techniques can be sodium fluoride for blood, and anticoagulants such as sodium citrate, potassium oxalate, EDTA and heparin are used in different cases depending upon the availability. The preservatives of the viscera are again very important in all cases of poisoning, but acid poisoning except carbolic acid. And the preservative used is saturated salt solution. And then for acid poisoning, except carbolic acid poisoning, rectified spirit is used. Then carbon monoxide poisoning, a layer of paraffin to prevent the escape of the gas. So this is a very logical use of the paraffin. And all cases of poisoning, including alcohol poisoning for blood, potassium oxalate, and sodium fluoride is used as a preservative in these cases. So in this section, basically, we dealt with the sample collection. How are we going to take it? What are the preservatives used in different instances? It is discussed over here. That is the end of this section. Thank you for watching sqadia.com.